That was a great session, and I think we'll be talking about that for some time to come, but also not just talking. It looks like we may have some action to come from that, so that's great. Can I have the innovation grant slides, please? Thank you very much. So it's my honor and pleasure to be involved in the Innovation Grant Award this year. This is the first of a kind, and the focus is on immunotherapy. We know that there have been 2 recent practice-changing studies which have shown an improvement with overall survival with the addition of immunotherapy, either durvalumab or pembrolizumab, to the chemotherapy backbone. We know that this reduces the chance of patients dying from the disease by 20 to 25%. And more than that, 1 in 4 patients derive a prolonged benefit with disease control over 2, and you'll see from posters at this meeting, even 3 years. However, in time, the cholangiocarcinoma very often shows disease progression, on average between 6.5 to 7 months later. And at this point, the cancer has then become resistant. So one of the questions that we're trying to raise is, how does secondary resistance develop, and how can this be overcome? Does it give us any new avenues of therapy? So against that background, this is a brand new initiative for the foundation. We thank the Bachrach Family Foundation for making this possible by generous donation to allow us to launch our first innovation grant. We hope this is going to be one of many, and this is different to the research fellowship grants, which tend to be small seed grants. These are larger grants, which are anchored in collaboration across institutions and disciplines. So following the award of the grant to the foundation, we went through a very stringent process of inviting letters of intent. We received 12 initial letters of intent. That included 5 US institutions, 10 countries overall, and 2 industry partners. Following review by the SMAB chairs and myself, 4 applications were invited for a full application, and these were received at the beginning of March. And since then, we've had a grant review meeting and a grant award meeting. I'd like to take a little bit of time just to thank the review panel. You'll see here that these are experts in the field of immune biology. So many of these people have got labs which are trying to address the question maybe in other cancers. And you can see the distribution of people from Harvard, Mayo, OHSU, Penn, once again, Harvard, Penn. And I'd like to thank Ben Stanger in particular, who chaired the review panel. I think for me, it was a fantastic opportunity to meet all of these great people, really hear their thoughts on the applications. And they were all mostly outside of cholangiocarcinoma, so they were really able to understand the science and the biology of the applications and how then this applied to our disease. I'd like to also thank Donald Kopok, who was the patient advocate who reviewed this. And this is the list of the 4 finalists and the institutions. You can see here that one was from US, one was a multi-center application from multiple sites in Italy. There was a transatlantic application from Canada, London and Dublin, as well as multi-center one, Johns Hopkins and Emory, together with Compass Therapeutics. I won't read all the individual titles, but I'll move on to the grant awardees. I'm delighted to announce that the 2024 Innovation Grant is awarded to Katie Kelly and Tim Gretton from UCSF and NCI respectively for their proposal entitled Molecular and Immunological Mechanisms of Acquired Resistance to First-Line Chemoimmunotherapy in Cholangiocarcinoma. Congratulations to Katie and Tim. I'd like to invite both Katie and Tim up to the podium together with Gordon to give us a very big glimpse into what their proposal is, and I'm sure you'll be hearing much more about this in next year's program. So thank you so much.
We have a couple slides to show, if those could be put up. So while we're getting the slides posted, I'll thank on behalf of all of our team members, the Cholangiocarcinoma Foundation for launching this innovation, the Bachrach family for funding it, and for this enormous, let's see. Yep, there we go, perfect. That was the right slide. So we put together a proposal entitled Molecular and Immunologic Mechanisms of Acquired Resistance to Chemoimmunotherapy in Cholangiocarcinoma. And just to share the call here was to identify factors contributing to acquired resistance in order to inform clinical trial paradigms and optimize the application of immunotherapy with more a precision medicine approach, especially in the context of resistance. And so, one of the challenges here is that we haven't really defined primary resistance to chemotherapy from an immunologic perspective yet in cholangiocarcinoma as well. And so we wanted to come at this with a team approach of different skill sets. And so I'm a clinician, as all of you know, a clinical trialist predominantly, but I have shown here a picture of team members that have very complementary skill sets from really one of our early career immunology, MD, PhD, translational scientist leaders, Dr. Bridget Keenan, who is at UCSF and would be here today, but she's at a memorial, unfortunately, and sends her regrets, but will definitely be here next year. My colleague and clinical partner and lab-based scientist, Dr. John Gordon from UCSF, Dr. Tim Gretton, who is a lab-based immunology researcher at the NCI, Dr. Huat Lim, who is a computational biologist and a hepatobiliary clinician and early career researcher at UCSF, and we're really excited to have him joining our team and also the Cholangiocarcinoma Foundation community. And from Dr. Gretton's team, Dr. Reda Benabarak, I think I got that right, who is a PhD immunology researcher as well. And so with these different skill sets, this is sort of a schema which shows the initial approach here. And again, I mentioned a minute ago that one of the challenges is not having a clear idea of what primary resistance looks like from an immunologic perspective, just to chemotherapy alone. And so we divided our resources into two existing cohorts. We have with Tim's prior immunology and immunotherapy trials at NCI and our prior immunotherapy trials at UCSF, we have collected PBMC samples from over 60 patients who have had primary resistance on chemotherapy and then gone on to get different types of immunotherapy. So we're using that as our retrospective cohort to define what those PBMCs look like in primary resistance, and then assembling a prospective cohort, samples of which we already have some collected from patients who are getting first-line chemoimmunotherapy. So we can compare what resistance looks like from chemotherapy alone to sequentially on immunotherapy. So the first aim is really an immunology aim, and the part that deals with patients in the clinic is to collect PBMC samples, both the previously collected old ones from patients on chemotherapy alone to the current ones in patients on first-line chemoimmunotherapy. And my colleague Bridget Keenan in Cancer Immunotherapy Program at UCSF will be profiling those PBMCs with site-seq and single cell to really define the populations at hand. And our primary hypothesis is that there may be myeloid mechanisms that drive resistance to immunotherapy both alone, but also in the setting of primary first-line chemoimmunotherapy. And then we'll take the findings from the human samples, and then I'll turn over to Tim so he can describe what he'll do in the lab at the NCI.
So first, I also obviously want, to thank the Cholangio Foundation and everybody else, for this award. It's truly an honor. And before I tell you about the mouse study, I think there's a few important points I want to make, what really reflects what CCF does here. This group that is working together here, has actually worked together before. And what we do here is, we basically foster collaboration. We have people with different expertise, younger and older, male and female, work, with the aim, to improve outcome here for patients. And that's, what we're trying with this. Now, let me tell you, and actually, I wanted to thank that the CCF is also having a whole day yesterday on basic science, and where I was able to show some of those studies where we actually immediately take data that we generate in our patients and then test this functionally in mice, because only there we can really figure out, why something works or doesn't work. And that's what we're doing here. We have the experience, we have the models to test this, and we can basically treat our mice with the same type of drugs that we use in our patients, and thereby, take the findings that we obtain initially in patients and then test this in mice and see how we can overcome this. Thank you.
Thanks. And I'm John Gordon. I'm a medical oncologist at UCSF, as Katie pointed out. And I'm speaking on behalf of the co-Is as a whole who are working on the tumor contribution. So one of the questions that my lab focuses on in cholangiocarcinoma and other malignancies is when we look at combination immune therapies, whether they be chemoimmunotherapy, as we're talking about here, or, for example, antivascular immune therapy combinations, as we see in other tumors, what is the tumor doing? How is the tumor contributing to the outcome? Because we know that with only rare exceptions, immune therapy on its own is not active for cholangiocarcinoma, and yet we have these data suggesting that immune therapy with chemotherapy combinations are active. And so then we have to infer that the effect of the chemotherapy on the tumor is contributing to the overall benefit. And so we're going to use a variety of methods using both tumor cell culture and using machine learning strategies to link tumor cell culture results that we obtain to large both patient and cell line data sets to identify what is the picture of a response to gemcitabine plus cisplatin, and how do we lose those features as tumor cell models and therefore by extension patients become resistant, in the hope that we can learn what's missing as we start to lose control over these tumors and try to replace it for patients going forward.
Thank you. We don't need to wish them luck. We know that this is in very capable hands. They have a fantastic track record of delivering. Ladies and gentlemen, your 2024 Innovation Grant Award winners.